semi-conservative mode of replication pattern. The word I can use only for most of the species, but not for all species. We know very well the conservative mode of DNA replication is also possible. I'm not understanding. You're saying semi-conservative, conservative. What is this? I'm not understanding anything. Can you explain to me in brief? Yes, of course. The double-stranded DNA. Can you see this place? The double-stranded DNA is getting unwinded, and once the double-stranded DNA is getting unwinded, by using the single-strand DNA, a new DNA strand is synthesized. A new DNA strand is synthesized. This is the term, or this is the process. We can bring it under semi-conservative mode of DNA replication. Whereas the conservative mode of DNA replication, if you're talking about the bacterial DNA or the mitochondria, bacteria is not having mitochondria. So bacteria cytoplasm is having circular DNA, which get segregated into two in number, not completely, partially, okay, like this, and then a new DNA is formed around the separated plasmid DNA, right? So conservative mode, I can say only for the circular DNA. Semi-conservative mode, I can say for double-stranded DNA. Like this, we can change the concept in detail. Now we are coming for the next segment is in the replication process. What happens exactly here is we know very well Watson Crick gave the double helical structure of the DNA. So this proposal leads to the idea of semi-conservative DNA replication. So we know very well the DNA strand separates and acts as a template for the synthesis of new complementary strand. So the new complementary strand synthesis, just only I narrated before going to the session, that is called a semi-conservative mode of DNA replication, right? Semi-conservative mode of DNA replication, right? So now we are entering into the next important concept, Matthew Meslin and Frederick Stoll experiment. This Matthew Meslin and Frederick Stoll experiment, in this we are using the ammonium chloride, which is having the 15N. 15N and 14N, of course, is light radiative isotopes. These light radiative isotopes, 15N, 4N, indicates the light radiative isotope. So we know very well about the 14N and we know very well about the 15N. So both the nitrogen are used by this bacteria. As a result, the corresponding DNA is formed, only rich in 14N DNA, which is having less white than the DNA which is having the 15N. So we know very well the DNA requires nitrogen. So I am growing the bacteria rich in the nutrition medium having the 14 nitrogen. I am growing the bacteria rich in 15N nitrogen. 15N nitrogen. So some bacteria are having 14N nitrogen, some bacteria are having 15N nitrogen, right? So the bacteria which is having 15N have much weight than the 14N DNA, right? So here when we are doing this type of semi-conservative mode of DNA replication practice, in terms of examining the way how the E. coli DNA is replicated, for that to understand this double-stranded DNA is getting duplicated into two after getting segregation, and by using the parental DNA, new DNA strand or daughter DNA strand is formed by using the parental DNA. So by using the parental DNA, new daughter DNA strand is formed. New daughter DNA strand is formed by using the parental DNA. This is the message we need to understand when we are discussing about the full name is Matthew Meslin and Frederick Stoll, right? But here we given only Meslin and Stoll, enough. Now we are coming for separating the DNA of different bacteria. We are using cesium chloride, which is taken into the centrifuge tube. This cesium chloride is inert to the 14N DNA and 15N DNA, and this gives a very good density gradient for appropriate separation of the radiolabeled DNA in the different area that will be compared to understand the semi-conservative mode of DNA replication. Now, if you are going for the detailed explanation that we already narrated for you, okay, we are growing one bacteria only in the 15N nitrogen, and we are growing the same bacteria in the 15N medium for other 20 minutes. When I'm keeping on changing the nutritional medium, okay, after some time, I'll be having bacteria with only 14N DNA, bacteria having 14N and 15N DNA, bacteria having only 15N DNA, bacteria having only 14N, bacteria having only 14N, bacteria having only 15N, bacteria having 14N and 15N, bacteria having for both 14N and 15N, right? So that is a message we need to understand. Now we are coming for the mechanism and enzymes involved in it. Before that, we'll go for the questions. Okay, the questions in 2023, this question was asked. The first question is exactly the replication process. Now only we discussed. Okay, Matthew Mesler and Frederick Stoll experiment explaining semi-conservative mode of DNA replication. Like this, we told already that is narrated under this particular subheading, Matthew Mesler experiment, right? So that narration should be here for this question, and this question belongs to last chapter, principle of inheritance, what we told in the last class. So in either question, two different chapter questions are coming, right? Can you see this in the view for the exam? Can you see this? This is the way the answer script should be, point by point, with a lot of subheadings. We told many number of time in many different type of classes of various curriculum. Okay, the point representation should be in this way with a lot of subheadings, right? So now we are entering into the next question. Is nothing but if you're talking about the bacteria transfer to 14N medium and sample every 20 minutes. Bacteria grows in 15N nutrition medium for many generations. So in which sequence this all this 5 should be arranged? This is the sequence. First 14N, and then we are growing the bacteria in the 15N nutrition medium. And then bacteria contains either all 14N and all hybrid DNA, right? So the 15N DNA amount has reduced at the end of the stage, right? So this is the way the sequence should be arranged. So I think so students can understand this message, right? This explanation you should understand, right? So subjective questions we discussed, assessment reasoning questions we discussed, case-based studies we discussed. Now we'll go to the question number 3. In 2018 board paper, they asked the dual purpose served by deoxyribonucleotide triphosphate. So of course, one is energy. ATP is serving as an energy molecule, right? So again, back I'm repeating, ATP is serving as an energy molecule. And beside that, it is a skeleton or an ingredients or a brick of the DNA. So that is the thing answer is given in this phase. So understand this, the dual behavior sense or the dual purpose of the DNP. So deoxynucleotide triphosphate message. This is the way you are understanding. Any one of you are having any doubt in this place, please let me know. So now we are entering the fourth question. In this fourth question, how was the RNA world hypothesis contribute to our understanding of the transition of non-life to the life on the earth? Discuss the potential scenario for the origin of the RNA-based life. If this is a question, so the answer should supposed to be like this. You know very well, here the RNA ingredients given the information about how our ancestors existed, how our ancestors originated, how our ancestors interacted with each other, how our ancestors accompanied themselves along with the various other species and with the environmental setup. So that information is explained by the RNA ingredients. So some organic molecule, including amino acids and nucleotide, or probiotic conditions. Okay. So RNA molecule assembled from these building blocks, possibly facilitated by the environmental factors. See, we are explaining Big Bang theory, the way how the earth is originated. The same way how the RNA is originated because of the addition of one ribonucleotide along with the other ribonucleotide, right? So now we are coming for the next phase explanation is ribosome is an enzyme. We all know very well that does RNA maturation process, right? So now we are coming for the next phase of explanation is in the replication process. We all know very well. I already told you bacteria does two type of DNA replication with circular DNA, conservative, with linear DNA, semi-conservative. Be clear to take the message, right? Now we are entering into the related videos. Whenever you are having time, please see this 3D video. We specifically posted for you for better understanding of this concept. Examples, worked examples, the difference between the leading and lagging strand in the DNA replication. So, leading and lagging strand replication, we all know very well. The lagging strand denotes discontinuous strand, and leading strand denotes continuous strand, which is because of the n number of Okazaki fragment or the primer. So the n number of primer, which is formed with the lagging strand, made the lagging strand DNA. Again, like I'm repeating. Okay. So if you're talking about so lagging strand and leading strand. Again, like I'm repeating. One is called as a leading strand. Okay, another is called what? Lagging strand, like this I already told to you. So you know what is the function of the leading strand, and you know what is the function of the lagging strand. Even though it is a lagging strand, discontinuous strand, it get irritated after some time in the normal process, whereas the leading strand will remain as such. Okay, so this possibility is because of the many number of primer when the lagging strand is getting synthesized, but that is not the case in the case of leading strand. Like this, we need to understand the message. Now we are coming for the significance in the origin of the DNA replication process. So we know very well the origin of replication is termed with many important terminology, not only origin of replication. So we can say as hotspot. So either you say ORI.C, or either you say hotspot, or either you say replicant. So either you say replicant, not recon. Recon meaning is different. Replicant or hotspot or ORI.C or origin of replication. So many terms they're using the different board paper. So you should be competent enough to understand, and you should say the answer more correctly, right? So now we are entering into the important tips of the exam. We all know very well, of course. Terminology, DNA polymerase, primer, Okazaki fragment, parental stand, daughter stand, leading stand, lagging stand, continuous stand, discontinuous stand. All these terms we need to align in our mind, and we need to bring the relevant concept of the same terms with the other associated sentence of the other terms which is in connection with this particular core term, right? So this type of practice is very, very important. In addition to this, we need to know the enzymes in which way it is participating in the various steps of this reaction, right? So this is about the tips for the exam. And in the case of presentation, we discussed a number of concepts, right? So now we are entering into the important replication mechanism. In this replication mechanism, okay, step by step we'll discuss. First, the DNA is getting unwinded because of the helicase enzyme, which is not given in the CBSE textbook. The double-stranded DNA is getting unwinded. The double-stranded DNA is getting unwinded in one specific segment because of the helicase enzyme. Again, back I'm repeating, because of the helicase enzyme, the double-stranded DNA is getting unwinded. Okay. After that, the DNA polymerase attaches to the DNA, and which DNA exactly? To the template DNA, in par with the template DNA. For example, in the template DNA of ASA, in the daughter DNA, the other nucleotides are added. Other nucleotides are added, right? So this is the parental DNA. This is the daughter DNA. So this is the way the parental DNA is formed. This is the way the daughter DNA is formed, right? Now we are coming for the next phase explanation is in this efficiency and speed. DNA polymerase must be highly efficient due to the need of the polymerization. So due to the need other polymerization. So this DNA polymerase, also called as DNA dependent DNA polymerase, which does new DNA synthesis, right? This many number of base pairs are there in the E. coli, and that completes the replication within 18 minutes. And this requires average approximately 2000 base pairs are formed per minute. Why this much fast? How come this type of speed replication we are experiencing in the case of bacterial DNA and that to E. coli DNA? Because the DNA is completely unpacked without histone. So there is no histone, there is no non-histone. There is a reason why the DNA is getting replicated into n number in a short time period, unlike the eukaryote. All right, this bacteria is a prokaryote. Now we know very well the energy resource for this replication to happen is ATP, is nothing but adenosine triphosphate. Because of this ATP, the new DNA is getting synthesized. This ATP is helping the DNA replication to happen. When the ATP is hydrolyzed, when the ATP is hydrolyzed, so now we are coming for the next phase of explanation. Is nothing but replication fork. When the double-stranded DNA is getting unwinded, what happens exactly here is when the double-stranded DNA is getting unwinded, you're getting the replication fork in this way. This by using this replication fork, the new DNA is synthesized. By using this replication fork, new DNA is synthesized. So the new DNA means one is the light color denotes what daughter DNA stand. One is of leading time, another is of lagging time. That means the lagging denotes discontinuous strand, unlike the continuous strand, which is on either side or same side or only one side. So these are all the different type of MCQs we are observing in the board, right? So by using the DNA or the panel type, the new daughter DNA is synthesized by using the DNA polymerase at the replication fork area. This is the way you need to understand while referring the board preparation points, which is exclusively given in your board university portal. Now, replication origin, we discussed with you. The replication origin, what we need to understand is basically the unwinded DNA. Okay, unwinded DNA will not promote replication. Winded DNA promotes replication. No, unwinded DNA promotes replication. Okay, so this unwinding of the DNA happens because of the DNA helicase. And once the unwinding happens, immediately the replication fork is created. This fork is responsible for making the new DNA strand. This is because of the DNA polymerase enzyme, also called as DNA dependent DNA polymerase. So this DNA dependent DNA polymerase, you can't see the slide, right? And you can't see the DNA helicase in the slide, right? So in the slide, we can't see the unwinded DNA will remain as unwinded because of a single standard binding protein not there in the slide. So being a teacher, when we are taking the class, so we should have a number of preparation before entering the class. So like this, you also should prepare in this revision class so that you will tap the difficulties very faster for your better board preparation, right? The single stranded binding protein keeps the unwinded DNA apart so that this unwinded DNA is taken for the new DNA strand synthesis, right? So now we are coming for the next important transcription unit. For that, we need to know some question practice. First, we'll go for some worked examples. In the worked examples, can you see here, when does the DNA replication typically happens during the cell cycle? Okay, typically happens during the cell cycle. DNA replication in eukaryote typically happens in the synthetic phase of the cell cycle. Synthetic phase means S phase, which is a part of the interphase in the cell cycle where interphase of the cell cycle happens exactly where in the cell cycle only interphase is there or any other mitosis is also there. So before the mitosis, interphase is happening. For example, you are having birthday function. Before birthday function, being a parent, your parents are preparing the quite a good amount of snacks, food varieties, arrangements, everything they are doing to have a effective get together with your relatives and friends. The same way, before mitosis interface happens, this mitosis is bringing the Before mitosis, which happens? Interphase happens. The part of the interface is synthetic phase. So the synthetic phase of the interface is a preparatory phase before mitosis. Before mitosis, right? So how does the DNA replication in prokaryote? It happens like this. Whenever you're asking this question, um, here you know very well. Yeah, the prokaryote DNA replication happens very faster. Here, and second thing is in the prokaryotic DNA replication, the origin of replication is only one. Whereas in the case of eukaryote, the origin of replication is multiple, right? So single origin of replication, multiple origin of replication. Okay. Even though the eukaryotic DNA replication happens slowly, in multiple place, DNA replication happens, right? What is the role of DNA ligase in the case of DNA replication? You know very well. The DNA ligase establishes the bond between the two nucleotide. We know very well the bond name is called the phosphodiester bond. We should be highly specific when we are saying which bond it exactly establishes. It establishes phosphodiester bond. Who establishes phosphodiester bond? Who establishes phosphodiester bond? As I already told to you, the which one establishes? DNA ligase. Phosphodiester bond. Again, back I'm repeating. Phosphodiester bond is a bond which is formed between the two nucleotides of the DNA. This is a message we need to understand.